Bismillahir Rahmanir Rahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we will start our MCQs lectures from 76. These are actually the MCQs that are related to the chapter number 3, and we already talked in the 3 video lecture. We already talked 25 MCQs respectively. In the previous videos lecture we already talked the two chapter MCQs as well. So if you miss these video lecture, it is present into my channel, or there will be also a link in the description. You can check out it. Now we will start from the MCQ 76 and will end to MCQs 105. So let's start. The 76th MCQ says that on Macon key media E. coli forms E. coli forms pink color colonies. C is the correct option. Chromium diphtheria required what? It mostly required potassium trailer medium. C is the correct option. Culture media for mycobacterium. So for mycobacterium tuberculosis, the culture medium is LJ medium is the correct option. 79 MCQs that Lepria bacillus is the best culture on which? So it is more the best culture of actually food pad of mice. B is the correct option. Culture medium for the Clostridia species. So for Clostridia species, the culture medium is Robertson cocked meat medium. C is the correct option. Clostridia welchi is a positive for what? It is mostly for positive for the Nilgar test. B is the correct option. 82 MCQ says that the Nilgar's reaction detect what? Nilgar reaction mostly detect the lectinase. C is the correct option. MCQs 83 says that the incubation period of the Clostridium welchi. So the incubation period for the Clostridium welchi is 8 to 12 hours is the correct option. MCQs 84 says that the average incubation period of tetanus is what? So for the tetanus, the average incubation period is 7 to 10 days. B is the correct option. Then we have MCQs number 85. The salt agar is used for, it is mostly used for the Staphylococcus aureus. B is the correct option. MCQs 86 says that the culture medium for the Leishmania. So the Leishmania, the culture medium is NNN medium. So B is the correct option. A simple asexual spore which developed by budding is known as blastospores. B is the correct option. Culture medium used for the fungus. So for the fungus we mostly use subbroad medium. A is the correct option. Nutrient medium is also used, nutrient broth agar, but the best medium is the subbroad medium is the correct option. MCQs number 89 says sterilization of fermentation equipment. The method is followed by chemically heating and radiation. So D is the correct option. All of the above. List below are substances which acid by the organism mentioned in A to E. Now these are actually the specific things, means the crystal violet, ampicillin, plaque vaccine, ripencid. These are actually the numberings, and it should be a cross match with the correct option. So the first is crystal violet. So for the crystal violet, it is mostly best for the it is actually best for the Lactobacillus aureus. So D is the correct option. For 1, D is the correct option. For 2, ampicillin IP. So the ampicillin IP is actually, it is used for mostly Microsporus luteus. C is the correct option. Then 4 says that 3 says that plaque vaccine. So plaque vaccine is actually the Pastulura pestis, means it is used for the Pastulura pestis is the best cross match for the 3. Then rifampicin. So rifampicin is actually Lactobacillus aureus, it is used to. Now 91, if we see the matching of the 91, so it says that Lysol. So the correct cross match for the Lysol is actually C, solution of cerol with soap. Then we have black fluids. So for the black fluids, the correct is higher boiling fraction for acid. A. For white fluid, the correct option is prepared from refined far acid. So B, for the 3B is the correct. For 4, iodophores. What is iodophores? So iodophores are mostly iodine combined with complexes, so E is the correct. 92. So for 92, Shika test. So Shik test is mostly used for diphtheria toxin C. Okay. Montauk's test is used for the tuberculosis A. Then we have sterility test. So sterility test is mostly used for detecting extensive microorganism B. So for 4, potency test, so for this the correct option is E, uselessness immunological product. 93, cross match says that glass syringe. So for glass syringe, the correct option is glass syringe. The correct option is dry heat C. Then we have disposable instrument. So disposable instrument is usually G, radiation. So D is the correct option. Respiratory part. What is respiratory part? So remember guys, respiratory part is actually the chicken pox in children. Okay. Dialysis machine is what? So dialysis machine is actually chemical. B is the correct option. Number 94. The item listed from A to D can be identified by the test given below. So Combs test, it is mostly used for the identification of non-agglutinant body. D is the correct option. Coagulase test, so coagulase test is obviously it is for the staphylococcus. So virulent staphylococcus aureus, B is the correct option. Then we have D, pneumonia can be cultivated in. So D, pneumonia is cultivated in chocolate agar and bread, serum broth. So E is the correct option, all of these. Okay, D, pneumonia can be identified. So it can be identified by microscopic culture. D is the correct option, all of the above. MCQs number 97 says that the diagnosis of the tuberculosis is carried out. So it is mostly diagnosed by Elmelter. So the correct option is E, all of these. MCQs number 98 says that the size of the virus can be determined by which? So it is mostly identified by the micrography. A is the correct option. More identified by micrography. It is also identified by ultracentrifuge at a high speed. So D is the correct option, all of the above, all of these. Differential staining of bacterial spores. So for the bacterial spore we mostly used Maillard staining, which is actually differential staining. 100 MCQ said that electron microscope study not help in identifying the section of the bacterial spore, but it is also used for the capsule as well. So C is the correct option. 101 MCQ says the Wilson and Bélier bismuth sulfate medium is used for the growth of what? So it is mostly used for the growth of Salmonella typhi is the correct option. MCQs 102 says that which rickettsia can be grow on blood agar medium. So lactobacillus is actually grow on the culture media, blood agar media. Okay, so A is the correct option. So that's all about the MCQs of chapter number 3, and I hope this video is helpful for you. Inshallah, in the upcoming videos lecture, inshallah in the next video lecture, we will start our MCQs of the chapter number 4 that is related to general properties of the microorganism. So that's all about the MCQs related to chapter number 3. I hope this video has helped you. Thank you so much for watching.